The night folds softly around you, and you find yourself sinking into a quiet warmth, the kind that settles behind your eyes when sleep is near, but memory still flickers. Your breath slows, steady like waves curling at the edge of thought. And in that space, just before dreams, you begin to drift into something older than dreaming. Something buried not in stories, but in cells. You are not made of stardust alone. You are also built from a language. A 4-letter language written in a spiral so small that it fits inside every cell of your body. This language is older than civilization, older than fire, older than breath. And yet, it is still speaking inside you right now. The alphabet of life is not spoken with a tongue. It is whispered through molecules, and for adenine, T for thymine, G for guanine, C for cytosine, 4 letters, 4 chemical shapes. But with them, life composes its symphony. Imagine for a moment that you could shrink yourself down, not just small, but impossibly small, and enter a single human cell. The world inside would not be quiet. It would shimmer with movement, hum with electric intent. And near the center, wrapped tightly in a coil like a sleeping serpent, you'd find the DNA-deoxyribonucleic acid twisted into its famous double helix, like 2 climbing vines wrapped around each other in a silent embrace. You float closer, weightless. Each rung of the spiral is a pair of letters with T, G, with psi linked like lovers who never part. These pairs repeat again and again in patterns that build bodies, memories, laughter, skin. They determine whether your eyes reflect the sky or the earth, whether your blood flows fast or slow, whether a child hums the same lullabies you once heard. This is not a book you read. It is a book you are taught, and it is long, so very long. If you unwound all the DNA in your body, every copy in every cell, it would stretch from here to the moon and back ... hundreds of times. Yet all that instruction fits inside something too. Small to see. Your cells are like libraries, and DNA is the language they've memorized. Each gene, a small chapter, tells the body how to grow, when to divide, what color, what shape, what function. Most of these instructions hum along unnoticed, repeating like background music. But some, small though they are, can shape your entire fate. A single switch in one letter, just one misplaced base, can mean the difference between ease and struggle, between health and suffering. A single typo in your genetic script can echo through every breath you take. Still, in most moments, your cells correct these errors. Faithful scribes with a patience we rarely notice. You inherited this code, half from one parent, half from another. A collage of lineage, memory, evolution, stretching back through time, through ancestors, through hominids, through creatures that once swam blind in warm oceans. And somewhere deep within you, echoes still stir from those ancient shapes. Yet your code is uniquely yours, never written the same way before, never again exactly repeated. You are a living draft, an unrepeatable edition of a story that took eons to write. But now, something has changed. For the first time in history, we are learning how to write in the margins of that sacred script. Not just read, not just observe, but edit. To change what was written, to fix a letter, rewrite a sentence, alter the ending before it arrives. This is no longer the realm of myth, no longer just gods and ancient stories molding clay into people. Now it is scientists in quiet labs, holding pipettes like quills, using tools as old as bacteria to reshape what life means. And the thought stirs something deep in you, a mix of awe and uncertainty. You think of music, how each gene is like a note in a long, elaborate melody. If we change one note, does the tune improve or vanish? If we change many, does it stay true or become something unrecognizable? And who decides what version is best? Still, there is wonder in the idea. A future where children are born free from inherited pain. A future where the flaws of biology do not dictate destiny. A future where our bodies are not just inherited, but chosen. You begin to realize DNA is not just a blueprint. It is also a memory of survival, a memory of choices made by millions of lives before yours. And now we hold the possibility to shape that memory, to preserve it or rewrite it. But how do we edit something so ancient, so precise? The answer lies in a molecule borrowed from the smallest of creatures, a tool first found in the immune systems of bacteria. It waits quietly in your mind now, like a page waiting to be turned. A molecular scalpel with a name that sounds more like science fiction than sciencefact.crispr.clustered. Regularly interspaced short palindromic repeats. The name is heavy, but the idea is delicate. A way to find a specific phrase in the genome and change it. Not just delete, but correct. Replace. Improve. CRISPR is not a blunt force. It is precise, elegant, like trimming a single leaf from a tree while leaving the forest untouched. You close your eyes and imagine this, a cell floating gently, soft and translucent like a jellyfish under moonlight. Inside, a CRISPR tool glides along the DNA strand, searching for a phrase it recognizes. When it finds it, it pauses, then cuts, quietly without pain, and the cell, as if knowing what to do next, begins to repair itself. This time using a new instruction. It is hard to believe something so simple can hold such power, and yet it does. With CRISPR, we now whisper into the genetic script, rewriting not only what is, but what will be. We are still learning the tone, the balance, the rightness of each stroke. But the pen is now in our hands. You feel the weight of it. And also the wonder. There's something profoundly intimate about this moment in science. To touch the same. Letters that shaped your hands, your thoughts, your heartbeat. To begin to understand their rhythm not as destiny, but as possibility. You wonder what your own code might reveal if it were read like a poem. Would it speak of strength you didn't know you carried? Would it reveal your quiet gifts, your hidden resilience? Would it show your place in a chain of countless lives, each one humming with those same 4 letters? You breathe in slowly, and in your chest, trillions of cells continue their silent work, copying, reading, translating your script into everything you are. Nothing has changed. And yet, everything has stopped because now we hold the tools to write in life's deepest language. And so you ask yourself, not with urgency, but with a soft, lingering wonder, if we can touch the code of life, how do we choose what to say next? Asterisk you. Return to the cell you visited before, glowing, translucent orbs suspended in the velvet dark. Inside, the spiral staircase of DNA still turns, whispering its ancient song in bass pairs and loops. But tonight, you are not only here to listen. Tonight, you follow a tool dot. It's not a blade, not in the traditional sense. It carries no weight, no edge, and yet, in its silence, it can rewrite life itself. CRISPR. The name echoes softly in your thoughts, like the click of beads falling in rhythm clustered, regularly interspaced short palindromic repeats. It sounds complicated, and it is. But in truth, CRISPR began not in human hands, but in bacterial bodies, tiny organisms learning to survive in a dangerous world. Bacteria, too, have enemies. Chief among them, viruses. For eons, microbes fought an invisible war. In a mere struggle, they evolved a clever defense. When a virus invaded, the bacteria saved a snippet of its code, tucked it into its own DNA like a warning label. If the virus returned, the bacteria recognized the intruder's code and cut it apart with a guiding molecule and a pair of molecular scissors. That ancient defense system is what we now call CRISPR. Born from survival, it has been borrowed by scientists and transformed into a tool not just for defense, but for delicate revision. Tonight, you float with it. You watch as a guiding molecule called RNA drifts toward a strand of DNA like a song looking for its harmony. This RNA is not guessing. It knows exactly where to go. It's been designed to match a specific genetic phrase, a sequence of letters in the DNA code. When it finds the match, it calls forth its partner. A protein named Cas9. Cas9 is the scalpel. You expect something loud, something explosive, but what you witness is gentler than breath. The protein latches onto the DNA, settles in, and makes a tiny cut. Just 2 strands, just a slice in the spiral. The cell pauses, notices the break, and as it always does, begins to repair. This time, it's handed a suggestion. Alongside the CRISPR tool, scientists deliver a replacement sentence, a corrected version of the phrase just cut. And the cell, ever cooperative, folds in the new words, sealing them into its script as if they'd always been there. You are watching not a fight, but a collaboration. Nature and science, side by side. This is precision medicine. This is controlled poetry, and it is still so new. You drift through lab after lab, some real. Some imagine where researchers stand beneath cold lights and whisper to cells in tubes. Some use CRISPR to silence a faulty gene that causes blindness. Others insert healthy instructions into cells of a sick child, hoping to erase a legacy of pain. In plants, CRISPR can remove the need for pesticides. In animals, it can resist disease before birth. Each act is small. A phrase here, a sentence there, but each one echoes. There is something powerful in watching a tool that does not roar, but edits. Like a careful author revising a manuscript, CRISPR does not reinvent life, it refines it. And yet with every edit comes a question. Where does fixing end and designing begin? It is one thing to cure illness, but what about traits? What about preferences? What about changes not born from necessity, but from desire? From desire. You begin to sense how thin the line is between medicine and choice, but still, tonight you stay in the space of wonder. You return to the single cell floating gently in its glass dish. You watch CRISPR work. The molecular scalpel hovers, precise and unshaking. No chaos, no violence, only intention. Only the soft click of molecules rearranging their story. You remember the old way, how gene editing was once a slow, blunt process full of guesswork and risk. CRISPR changed that. It made the genome feel more like clay and less like stone. It offered not certainty, but possibility. And in doing so, it opened a new chapter for life itself. Imagine for a moment that every cell in your body is a word, and every tissue a sentence. CRISPR lets us edit those words mid-paragraph. Change one and the whole meaning may shift. Or remain the same, but stronger, cleaner, healed. That is the gift and the gravity of this tool. You think of all the ways it might unfold in the future, in the hands of doctors, of farmers, of parents, perhaps one day even in your own. But tools are only as gentle as those who wield them. So CRISPR waits, quiet and faithful inside the cells of mice and wheat and human embryos. It waits in the silence of possibility, asking not for permission, but for understanding. And you, drifting in the warmth of sleep, begin to wonder, what will we choose to change? The tool is in our hands now. The scalpel is sharp. The edits are possible. But the story is still ours to write. And just beyond this moment, in a softly glowing lab, something new is floating in a dish alive, potential untouched. Embryos like stars waiting for a future to be whispered into their code. Asterisk, the room is quiet, lit with the faintest gold, like the last hour of dusk before the stars appear. But here inside this lab, the stars are not above you. They are beneath glass. You peer into a petri dish. Inside it, suspended in warmth and light, float the tiniest beginnings of life embryos. Only a few cells wide, translucent and still. They do not yet have eyes or hearts or hands. But in their spiral code, each already holds a future. They shimmer like constellations in a miniature sky. And you are here not to wish upon them, but to wonder what shape their futures might take. This is where science meets intention. This is where design begins. You feel no rush, no cold precision, only the hush of curiosity pressing against your thoughts. What can we do now with the ability to edit the code before life unfolds? The question is not hypothetical anymore. With CRISPR, the process begins before birth, before implantation. Scientists extract the DNA of a fertilized egg, scan its sequence like a map, and look for mistakes, risks, vulnerabilities. In some cases, the errors are devastating, a mutation that guarantees a child will suffer. But what if, with a single quiet cut and paste, we could fix that? What if suffering could be prevented before it ever began? You see it clearly now, each embryo like a possibility held gently in time. None of them asked to be changed, but all of them carry potential for pain, for joy, for brilliance. For lives both ordinary and extraordinary. And still the question lingers, where do we draw the line? There's a line between therapy and enhancement. One is relief, the other ambition. Curing a disease is one thing. But what if we begin to choose the color of a child's eyes, the height of their frame, the shape of their smile, the speed of their thoughts? You imagine a parent leaning over a form not to register their child for school, but to select from a menu of possibilities. Health, yes, but also talents, IQ, creativity, athleticism. It feels like science fiction, but the science is already here. What we do with it is the fiction still unfolding. In one corner of the lab. You watch a researcher peer into a microscope, hands steady, breath calm. She's not playing God. She's studying the code of a child not yet born, searching for places where the story might run off track. She isn't trying to build a perfect human. She's trying to avoid the ache of a preventable fate. And yet even this act of compassion holds a quiet power. Because in changing a gene, we change everything downstream. The proteins it produces, the cells they build, the organs they shape, the life they touch. Change one letter and the tone of the symphony shifts. Still, the temptation grows. If we can fix one thing, why not fix two? You breathe deeply. The room feels heavier now. Not dark, but dense with the weight of responsibility. Because every choice made in a dish today echoes far beyond the walls of the lab. Into families, into generations, into society. The child edited for health may live longer, stronger, and in that change the course of history in tiny, invisible ways. And what about the others? The unedited? Will they still be enough? You begin to wonder if we are not just designing individuals, but designing expectations. The dream of a better human is not new. It's a dream as old as myth. The perfect child, the genius, the hero. But now that dream has molecules and machinery behind it. CRISPR does not dream, but it enables dreams to become instructions. The embryos in this dish cannot speak, but they carry a language that we have only just begun to understand. And in reading that language, we face a choice. Do we let it guide us gently, or do we try to rewrite its grammar? Outside, life continues as always. People are born with quirks and scars and brilliance and faults. They grow into their shapes like wildflowers bending toward whatever light they find. But here, in this dish, the wild is tamed. The randomness quieted. You wonder, will we lose something in our pursuit of perfection? Perhaps it's not the flaws we need to erase, but the fear of them. Still, you drift back to the beauty of the science. Because there is beauty here, the power to heal before hurt, the ability to give a child a life not defined by diagnosis, the hope of removing suffering not through miracle, but through careful, compassionate design. You think of a baby years from now breathing easily where once it could not, laughing, running, thriving because a scientist once paused beside a petri dish and made a change no eye could see. That child will never know the moment their story shifted. And maybe that's the point. We are shaping silent futures. The lab fades gently as you step away, your mind still wrapped around the light of the dish. The possibilities swirl gentle, uncertain, filled with questions you don't yet have to answer. But others already have. And as you begin to drift deeper into sleep, your thoughts follow two little girls, born into the world not as nature left them, but as human hands shape them. Born not just with DNA, but with history. You wonder, how did it feel to be the first? Asterisk, it began not with thunder, but with a whisper. A quiet announcement in a conference hall in Hong Kong, where one man stood before the world and said, "It has been done." The words rippled outward. Invisible and irreversible. Lulu and Nana were born in China in 2018, twin girls. Healthy, soft-skinned, wrapped in the quiet folds of infancy. But inside them, inside every cell, was a mark, a scar of intention. They were not born as nature had designed them. They were designed before they were born. The scientist, He Jiankui, had used CRISPR to delete a single gene from their DNA CCR5. This gene, when functional, can allow each ivy to enter the body's cells. By erasing it, he hoped to make the girls immune. A cure, written into their code. You imagine it now. A quiet lab, a petri dish, a needle barely visible. A moment when the scissors snipped something ancient. The spell of randomness broken, and from it, 2 lives, 2 girls, who did not choose this beginning, but were forever shaped by it. You breathe slowly. The world did not cheer Dot because this was not how it was meant to happen. The scientific community, prepared for careful, cautious steps, was jolted into alarm. Asterisk, the experiment had not passed peer review. It had not met the standards of transparency, safety, or ethics. It had been secret, and in the silence, it had leapt ahead of the world. Lulu and Nana were not theoretical. They were living proof that the line had already been crossed. You sit with this truth. These girls were not sick. They were not facing a deadly condition. They were made into an experiment not to heal them, but to test what was possible. And so the outcry came. Was it protection or risk? Was it love or ambition? Asterisk, the scientist who edited them was sentenced to prison. His research halted. The lab doors closed, but the story did not end, because Lulu and Nana are still out there. They are growing, aging, living, carrying in their DNA, a change no other human carries, and no one knows what comes next. That's what makes it different. This wasn't a cure given after birth. It was a rewrite that will echo through every cell in their bodies, and in their children's children, if they someday choose to have them. You wonder what it feels like to grow up knowing you were edited before you were named. That your life was the first of its kind. Do they know? Do they understand the weight? Or do they run through sunlight, laugh over crayons, dream of stars, never once suspecting that the world watches them from behind scientific eyes? You lean into the thought. They didn't consent. They didn't agree to be the first. And yet the silence around them is deafening. For their protection, their identities are hidden. We don't see their faces, but we see their shadows. They remind us of what science can do and what it must be careful not to do again. But they are more than symbols. They are children. Real, breathing, dreaming children. And in the heart of all this noise, you try to remember that. You picture them now. Walking along a beach, small feet pressing into wet sand. Waves rolling in beside them, slow and endless. Behind them, tiny footprints. A head open sky. They are not experiments. They are lives. You close your eyes and imagine a world where their stories not defined by the lab, but by their laughter, where the edits fade into the background of who they truly are. But still, the world watches. Every doctor, every policymaker, every hopeful parent who wonders if the next child might be stronger, faster, smarter. ICE-PR unlocked the door. Lulu and Nana stepped through it. And now we all stand on the threshold. Some see a miracle. Others see a mistake, but everyone agrees. This was the beginning. You exhale slowly, letting your thoughts drift like mist across the water. The girls are out there. And whatever your opinion, you hope they are safe. You hope they are happy. You hope their names, when someday they are spoken with gentleness. Because history does not always remember its pioneers kindly, but it remembers them completely. And Lulu and Nana will never be forgotten. Asterisk, you're floating in a soft silence, a space between imagination and reality, where science offers dreams with fine print attached. This is not about curing disease anymore. This is about potential. About a child born with sharper memory, clearer skin, stronger muscles. A body that never forgets how to heal itself. A brain that sees patterns faster than you ever could. You ask yourself, would you say no? Imagine the moment you're holding a tiny bundle of cells. The embryo is no larger than a grain of salt, but already it holds 1,000 futures. And someone asks you, would you like your child to be free from Alzheimer's? Of course, would you like them to be resistant to cancer? Absolutely. Would you like them to be smarter? Just a little? What about taller, more athletic, more symmetrical in the face? A voice that sings just a little sweeter? The line begins to blur. What begins as protection soon becomes preference. You think back to the beginning, CRISPR as a shield against suffering. But now it begins to look like a sculptor's tool, carving humans into ideals we haven't even agreed upon. What is intelligence? What is beauty? Who defines strength? Would we be chasing the same traits in every child until uniqueness fades into sameness? You sit with the discomfort because deep down we want the best for our children. That's the seed of all of this. But what if the best isn't what we think it is? What if compassion matters more than IQ? What if resilience grows from struggle, and the edits we make rob someone of that growth? You picture a world where enhancements are common, not illegal, not hidden, just expected, and suddenly a child born without edits is seen as incomplete. Would that child feel shame? Would their parents feel guilt? You think about the pressure, about the arms race it might create. One nation edits for speed, another for memory, another for obedience. Where does it stop? And who gets left behind? Because editing costs money, and money buys access, and access shapes opportunity. Will we build a future where the rich edit their children into perfection, and the rest of the world is expected to compete? You exhale slowly. It starts to feel less like a question of science and more like a question of justice. Of equality, of humility, because the more power we gain over biology, the more we must ask, do we know enough to wield it kindly? You sit in the stillness, watching possibility unfold like ripples in a lake. A child with enhanced vision paints galaxies. Another solves equations with impossible speed. Another climbs mountains without pain, never fearing disease, never fearing age. It's beautiful. But is it fair? You imagine a schoolyard. One child leaps effortlessly across the finish line. Another stumbles behind. Not because they didn't try, but because their DNA was never upgraded. How would you explain that? Would we still value effort? Would we still honor struggle? Or would we begin to see success as something pre-written, something purchased before birth? You breathe in deeply. The question is not whether we can make better humans. It's whether we know what better really means, and whether we've learned enough about our own hearts to avoid turning children into projects. Because a child is not a goal, not a checklist, not a reflection of our fears or fantasies. A child is a question, a spark, a song not yet sung. You lean back into this thought. If we edit for strength, do we risk losing gentleness? If we edit for brilliance, do we forget kindness? What if, in Shaping children to be more, we accidentally make them less curious, less forgiving, less themselves, because genes are not switches. They are webs. Tug one string and others quiver. We don't yet understand the full harmony of the human song. You return now, gently, to the question, should we? Should we edit for intelligence, beauty, or strength? And the answer is not clear. Not yet, because this is a mirror held to our values, and we are still deciding what we want to see reflected. You whisper one final truth to yourself. Just because we can sculpt the future doesn't mean we must shape it with hard edges. Sometimes the most beautiful things are the ones we never planned. Asterisk, you are floating again. Not in dreams this time, but in the quiet hallway between what could be and what must be done with care.cr. IcePR sounds clean. When you say it out loud, sharp, smart, simple, like a surgeon's tool in steady hands. But life is not a fixed machine. It is movement. It is mystery wrapped in motion. And when you try to cut something so alive, sometimes it doesn't heal the way you expect. You drift now into the story of risk. A child's DNA is edited to remove a disease, but in the process, something shifts. Something tiny, something unplanned, a single base pair changed elsewhere. A ripple that bends the music of the body. And suddenly, what was meant to save starts to harm. These are called off-target effects, errors, small cuts in the wrong places. It's like trying to fix a page in a book, but the ink bleeds onto the next one, changing the sentence you didn't mean to touch. You can't see it with the naked eye, but it's there. A gene that once worked perfectly now skips a beat. A protein folds just wrong. A system fails quietly. Sometimes not for years. Sometimes not until the child grows, until puberty hits, or stress appears, or 1,000 other unknowns awaken the change, and by then, the edit can't be undone. You begin to feel the weight of this. CRISPR is not evil. It's not reckless. But it is powerful, and power without full knowledge is a trembling hand. You. Think of your own body. How many systems hum beneath your skin? Blood, bone, nerve, memory, sleep. How they work together in ways we still don't understand. How changing one gene might help, but might also unravel a thread that keeps something else in balance. It's not that scientists are careless. They're not. They run tests. They use computers to model risks. They double check, triple check. But the human genome is not a single path. It is a forest, dense, alive, ancient. And you do not walk through an ancient forest with loud feet. You pause at the word mosaicism. It's when not all the cells are edited. Some remain unchanged. Some carry the fix. Some carry mistakes. A patchwork child. One body, many codes. This happens sometimes. And the child may not show symptoms right away. They may look healthy for years, until the patch begins to tear. Then there are unintended traits. What science calls pleiotropy. You change a gene for strong bones, but find it also affects the heart, or brain development, or the way emotions are regulated. The genome doesn't work in silos. It sings in chords. And if you change a single note, you may shift the entire song. You feel a chill, not from fear, but from awe. Awe at how complex we are, How much we've yet to learn. You remember that early experiments in animals sometimes had unexpected effects. Pigs born with better muscles but compromised immune systems. Mice made resistant to one disease, but suddenly vulnerable to others. CRISPR may be precise, but life is unpredictable. And then there's the biggest risk of all, germline editing. When changes don't stop with one child, but pass on to every child after them. A single decision echoed across generations. And if we get it wrong, the error lives on. Invisible at first. Silent. Until one day, it doesn't stay silent. You breathe slower now. The air is heavier here. You understand that every tool, no matter how powerful, comes with questions it cannot answer alone. And those questions are not only scientific, they are ethical, emotional, philosophical, because you are not editing a machine. You are shaping a person, and that person may never consent. They are born into a choice they never made. You wonder what it means to be responsible, not just as a scientist, but as a society. Can we slow down? Can we resist the urge to move fast, to race toward perfection before we even agree on what perfect means? You hope so. Because getting it wrong is not like spilling a glass of water. It is like planting a tree in poisoned soil. It may grow. It may bloom. But the roots, remember. So you sit in this quiet. You listen to the silence that comes after too much certainty. And you realize the question isn't, can we? It's, are we ready to live with the answers if we don't get the question right? You find yourself in a place without walls, only borders. Lines drawn in ink and silence. Invisible until you cross them. This is not a laboratory. It is not a nursery. It is not a hospital. It is a map. And every country, every culture has drawn its own rules around the question, what should we be allowed to change in a human being? You drift above continents now. Some glimmer with red lights. No, never, not yet. Others blink yellow, maybe, carefully, under watchful eyes. And if you pulse green, yes, but quietly. We won't talk about it. In the United Kingdom, there is cautious exploration. A regulatory body, the Human Fertilization and Embryology Authority, sets limits with trembling fingers. CRISPR may be used for research, but never for live births. Not yet. In the United States, there's no outright ban, but no federal funds may be used. A barrier made not of law, but of budget. Private labs still hum behind closed doors. Science doesn't sleep for politics. In China, there was a moment the world cannot forget. A scientist announced the birth of twin girls, edited embryos, made resistant to ACE. The outcry was swift. Condemnation thundered. The man was jailed, but the children were born. And they are growing now somewhere. Living symbols of a line. Crossed out. The laws came after. But the act had already echoed. You feel the tremble in that, because law follows footsteps. It really runs ahead. And science. Science is fast. You ask yourself, who has the right to decide when a line is too soon? Is it governments, ethics boards, doctors, parents, or the children yet unborn whose lives will carry the decisions made in silence? You imagine a couple in one country, desperate to edit out a genetic disease, but their homeland forbids it. So they travel to where the rules are looser, To where oversight is softer, it's called reproductive tourism. Hope flies across borders just as quickly as risk. Now imagine this. 2 children, born the same year. One in a place where editing was allowed, the other where it was not. One child has been designed for better health, stronger immunity, sharper cognition. The other was born untouched, and so begins the quiet divide, not of wealth, not of education, but of biology. You feel it now. The creeping possibility of a genetic elite, not from science fiction, from policy. Decisions made today. When rules differ, so do futures, and then come the corporations. Private clinics promising healthy children. Better outcomes, designer traits. Not yet eye color. Not yet height or intelligence. But the language is soft and persuasive. We're just reducing risk. We're giving your child a better chance. Why wouldn't you? And suddenly, what was once a rare option becomes an expectation. Subtle pressure builds, and choice becomes burden. If a parent can prevent disease but doesn't, are they irresponsible? If they choose not to enhance, are they holding their child back? These are not questions science can answer. These are questions that live in courtrooms, in parliaments, in living rooms late at night. They live in the space between can and should. You hover now above a conference hall. A global summit on genome. Editing. Voices rise from microphones. Debates unfold in many languages. Some argue for a global ban until we understand more. Others say the time is now. That delay costs lives. But beneath every argument is fear. Fear of the unknown. Fear of falling behind. Fear of losing control. And yet you also hear hope that we can come together. Hope that ethics can keep pace. Hope that no single country decides the future for all. You descend gently back into your thoughts. CRISPR is not the danger. CRISPR is the invitation. It is we humans who must decide the terms. You understand now that policy is not just paperwork. It is protection. It is promise. It is a way of saying we will walk carefully because we hold the future in our hands. Asterisk, you drift now into a quiet waiting room. Not in a hospital, but in a future clinic where hopes line up beside wallets. Family sits on one side. Their child holds a folder. Inside, genetic risk reports, family history, a recommendation for editing. Across the room, another child waits. Same age, same condition. But their parents cannot afford the consultation, let alone the treatment. This is where the story fractures. Science is the same. The tool is the same. But one family gets healing, the other gets hope deferred. You begin to feel it now, the pressure of inequality. Not in wages, not in schools, but in genes. The new currency is access. When insulin was discovered, it was meant to be shared. A miracle for the many. But even now, some still die for lack of it. So you ask, what will happen when editing the blueprint of life becomes something only some lives can afford? A price tag on prevention. A surcharge on survival. CRISPR is precise. But access to CRISPR is not in wealthy nations. Private companies begin to offer packages. Pre-implantation screening and editing, $50,000. Enhance immunity for an extra $20,000. Bundle discount with in vitro fertilization. They market it as a choice. But it's only a choice if you can pay. And so a new kind of hierarchy begins to form, not from effort, not from education, but from molecular advantage. You imagine a classroom. One child never gets sick. Another misses school from chronic illness. One reads faster. One struggles with cognitive delays. Neither chose their DNA, but now some DNA is chosen, and that choice is shaped by income. The divide grows deeper. Quieter, more permanent. You begin to understand. This is not just about ethics. It is about equity. Left unchecked, CRISPR could widen the very gaps we've spent centuries trying to close. It's not the technology that creates injustice, but the systems we build around it. Insurance companies begin to take notice. Will they charge more for unedited children? Will policies cover preventive editing? Will the unedited be labeled high risk? The questions blur. You walk through a future neighborhood. Two clinics side by side. One with polished glass, warm lights, offering full spectrum editing for every embryo. The other barely lit offers only diagnostics. You may qualify for assistance, a poster says. But funding is scarce. Waiting lists grow longer. Diseases that could be prevented are merely tracked. It feels like a betrayal of the very idea of progress. You hear the whisper of a protest march in your mind. Gene editing is a human right. Healthcare, not design or care. But slogans cannot rewrite policy, nor can they lower costs overnight. So you ask, can we imagine a world where CRISPR is free, public, global? Where every child, no matter where they are born, has the same chance at health? It would take cooperation, funding, commitment beyond patents and profit. It would mean remembering that a better world is not one where the best genes win, but where every life gets the same chance to begin. You float now above a globe of possibility. Some regions are bright with research and accessibility. Others flicker, waiting for funding, caught between hope and hardship. You want to fix it, but you are only a witness for now. Still, witnessing matters. It is the first step toward responsibility. You close your eyes and carry the weight of this truth gently. Without fairness, even miracles can become weapons. Without equity, even medicine becomes a gate. And without access, even the most brilliant future belongs only to a few. Asterisk, you return now to the double helix, spinning quietly in the dark. Each bass a note in a song that has been playing for billions of years. And into this melody, we now place a hand. We adjust a single key. But what if, in tuning the future, we strike a wrong chord? CRISPR is sharp, but not perfect. It cuts, but sometimes cuts too far. These are the off-target effects, mutations, and places we did not mean to touch. Like trimming a branch and accidentally slicing the trunk. You imagine a child edited for stronger immunity, but a year later a cancer appears. Not because of nature, but because of us. You imagine a generation born with precision edits. They look healthy. They grow, they learn, they love. But when they have children, a subtle change, a ghost in the gene appears in the next generation. We had no idea. Some risks are immediate. Others lie dormant, waiting silently across time. In the early days, a scientist edited a human embryo, not for disease, but for resistance to HIV. It was rushed, controversial, and criticized. But beyond the ethics, there was a question that haunted the world. Did he get it right? And even if he did. Should he have tried? You now imagine being that child. Born with edited DNA, the world watches you grow. Not for who you are, but for what you represent. Every illness, every quirk becomes a case study. Every emotion is examined like a result. You were supposed to be proof of progress, but what if you're just human? Asterisk the burden of a single experiment placed inside your skin. The deeper you go into this future, the more fragile it feels. Not because the science is wrong, but because the unknowns are enormous. Biology is not code. It does not compile cleanly. It grows, mutates, adapts. It resists being controlled. You think of nature's own mistakes, mutations that cause suffering, but also the mutations that led to art, to language, to love. So you wonder, what happens when we begin to remove all? Risk? What beauty might we lose by fearing imperfection? Perfection. You return now to the lab. A scientist stands before a glowing screen. An embryo flickers under the microscope. They hesitate. There is a typo in the code, one that might cause pain. Or one that might do nothing at all. Or worse, something we don't yet understand. They look to the data. It is promising, but not complete. And still, the parent signs the consent form. Hope outweighs doubt. Love demands action. The edit is made. Somewhere in the future, a life begins. Not knowing it was shaped by this moment of courage or hubris. This is not science fiction. It is near science. And it is already shaping policy, law, and fear. You drift now through a field of ghost edits, potential changes never made, mistakes narrowly avoided, and some that were not. It is sobering, but not paralyzing. Because with each mistake we learn. And the lesson is always this: we are not gods, we are gardeners. Tending a garden we barely understand, with tools still new in our hands. The key is not to freeze, nor to rush, but to walk slowly. To build systems of review, of consent, of compassion. To test in models, to listen to caution, to question ourselves endlessly. You think now of another child, untouched, unedited, and yet just as whole. Because sometimes what is natural is not broken. CRISPR may cure, but it may also complicate. The safest path is not to avoid it, but to approach it with humility. To admit what we do not know, to pause before every cut, to understand that in the song of life, even one wrong note can echo far. Asterisk, you stand now at the edge of possibility. Behind you, ancient DNA, mutations made in darkness, life born of chaos and chance. Before you, a garden not yet planted. You hold the seed. The editing tool rests beside you. Sharp, quiet, ready. C. Our ASPR is no longer science fiction. It is science. Made personal. Science that knocks on your front door, offering hope and asking for wisdom. Imagine a world where no child dies of genetic disease, where sickle cell and cystic fibrosis are memories whispered in history books. Where every embryo is gently screened, not for perfection, but for mercy. A world where blindness can be reversed, muscles strengthened, brains healed. This is not fantasy. It is beginning now, but imagine also another path. A future where the lines blur, where edits are made not for health, but for height, for intelligence, for skin tone or temperament, What begins as healing can drift toward design, and design is never neutral. It reflects culture, bias, economy. Who gets to choose? Who can afford to choose? You imagine a global divide, those who are enhanced and those who are not. The genetic haves and the have-nots, a world split not by effort or education, but by birthright purchased in a clinic. Unless we choose differently. Unless the future we build is shaped not by fear or profit, but by principle. By a deep, shared understanding that editing life must never erase dignity, and that humanity is not a puzzle to perfect, but a mosaic flawed and beautiful. You imagine councils, ethics boards that include not just scientists, but poets, not just doctors, but mothers. You see classrooms where children learn not only what DNA is, but why care matters more. You walk through a city of the future. Neon lights flicker. The air is clean. Hospitals are quiet not because people are perfect, but because many were healed before they were ever hurt. And yet the diversity remains. Quirks, imperfections, differences of mind and body. Not edited away, but understood, embraced. Because somewhere we remembered to be human is not to be flawless. It is to be kind. You imagine your own children or grandchildren asking what it was like when CRISPR was new. You smile and you say, we were afraid, but we were careful. We argued. We paused. We listened. And then maybe you say something simpler. We chose love, not control. The future may come quickly, but choice always begins now. You return one last time to the cell inside. The machinery hums. Cas9 glides along the strand. It finds its target. It prepares to cut. And you are the hand that decides whether to let it. You breathe. The future waits.